Welcome everyone to the Ultimate Zero Shortcut Masterclass. Today, we are taking a microscopic dive right into Prep 3 Science, Term 2, Unit 1. The mission here is super simple. We're breaking down every single detail, scientific law, and definition from your revision sheet so you can achieve absolute mastery. No shortcuts.
Absolutely. We aren't just going to sit here and memorize facts today. We are going to really understand the underlying mechanisms. I'll be right here to help visualize exactly how these chemical phenomena actually work in the real world. So let's just jump right in.
Okay, here's our strict chronological roadmap for the session. We're going to start with the fundamentals of chemical reactions, then dive into thermal decomposition and colors. After that, we'll decode the chemical activity series, master oxidation and reduction, analyze reaction speeds, and finally wrap up with catalysts and everyday applications. All right, Chapter 1, the fundamentals of chemical reactions. Let's get into it.
So before we start looking at test tubes and balancing equations, we really need to establish exactly why chemical reactions govern literally the entire world around us.
Right, because science isn't just confined to a lab, it's happening everywhere, all the time. Just think about burning gasoline in a car engine. That combustion reaction is what generates the power to actually make the car move. Or, look at a simple leaf. Plants are actively making their own food through photosynthesis, which is just a massive chemical reaction combining carbon dioxide and water. Even the medicines that heal us, the fertilizers that grow our food, the artificial fibers in the clothes we're wearing right now, they're all direct products of chemical reactions.
Which brings us to the exact academic definition. A chemical reaction is defined as the breaking up of the bonds in the reactant molecules and the formation of new bonds in the products or resultant molecules.
Think of it kind of like a set of Lego building blocks. You start with one structure, right? Those are your reactants. But to build something entirely new, your products, you first have to pull those old blocks apart. You're breaking those initial bonds before you can snap them back together into a brand new configuration.
Moving right along to Section 2, Thermal Decomposition and Colors. Now, this category is incredibly important for your final exams, mainly because it deals heavily with very visible physical changes, specifically shifting colors.
Exactly. Let's systematically walk through this data because it's crucial. When we apply heat, these compounds decompose, and the visual evidence is just striking. For example, red mercuric oxide decomposes to yield a silver precipitate of mercury. And what about our copper compounds? Well, blue copper hydroxide, green copper carbonate, and blue copper sulfate all decompose when heated to produce copper oxide, which turns a very distinctive black. Finally, white sodium nitrate decomposes into sodium nitrite, which has a very specific yellowish-white color. You absolutely want to memorize these color transitions. They are classic exam material for sure.
And beyond just test tubes, thermal decomposition literally saves lives. Which brings us to the airbag, defined as an inflatable bag folded inside the steering wheel in modern cars.
Right, and the engineering here is brilliant. When a crash occurs, an electric spark triggers the rapid chemical decomposition of a substance called sodium azide. In literally a fraction of a second, this reaction produces a massive amount of nitrogen gas. That gas inflates the bag and cushions the driver. It's just a perfect real-world application of rapid decomposition.
Section 3, the Chemical Activity Series. Okay, so we're moving into substitution reactions now, and the absolute rulebook for how elements swap places is the Chemical Activity Series, or CAS. It's defined as the arrangement of metals in a descending order according to the degree of their chemical activity.
A great way to think of this is like a leaderboard. The strongest, most active metals are sitting right there at the top. A metal can only kick out or substitute an element that sits below it on this leaderboard, and the further apart the two metals are in this series, the faster that substitution happens.
And this brings us to a super classic give reason exam question. Why does zinc react with dilute hydrochloric acid immediately, but copper shows absolutely no reaction? It all comes down to where they sit relative to hydrogen on our leaderboard. Zinc sits above hydrogen in the chemical activity series. That means it's more active and can easily substitute it, forming a salt and releasing hydrogen gas. Copper, however, sits below hydrogen. It just doesn't have the chemical strength to force hydrogen out of the acid, so no reaction happens. But there is a really tricky exception you have to watch out for. Give reason. Why does aluminum delay after zinc in the reaction with dilute acid, even though aluminum actually comes before zinc in the chemical activity series? Yeah, on paper, aluminum totally should react faster. But in reality, aluminum has this microscopic protective shield. There's a layer of aluminum oxide, Al2O3, right on the metal's surface.
So when you drop it in acid, the acid first has to literally eat through this oxide layer just to reach the pure aluminum underneath. That separation takes time, and that's exactly what causes the delay.
Now, let's move from simple to double substitution. This occurs when two salt solutions react to form two new salts, and this is typically accompanied by the formation of an insoluble precipitate that just doesn't dissolve in water. A really perfect example of this is mixing sodium chloride with silver nitrate. They basically swap partners, giving us sodium nitrate and a very specific visual cue, a solid white precipitate of silver chloride that just falls right to the bottom of the test tube.
All right, Section 4, Oxidation and Reduction Masterclass. We're now transitioning from those visible physical changes like the color shifts and precipitates we just talked about into the completely invisible, microscopic world of oxygen transfer and electron flow. Let's start with the traditional concept. Oxidation is a chemical process which causes the increase of the oxygen percentage or the decrease of the hydrogen percentage in a substance. Conversely, reduction causes the decrease of oxygen percentage or the increase of the hydrogen percentage. They are exact opposites. But you know, as science advanced, we realized these reactions can happen without oxygen being there at all, and that gave rise to the electronic concept. Here, oxidation is a chemical process where the atom loses an electron or more. Reduction is where an atom gains an electron or more. Just remember this rule of thumb. Metals tend to undergo oxidation by losing electrons, while nonmetals undergo reduction by gaining them.
Now, I need you to pay very, very close attention to this next point, because it is the absolute #1 pitfall for students on exams: the agents. The relationship here is completely inverse. The substance that undergoes oxidation is actually the reducing agent, because it loses electrons and gives them away. The substance that undergoes reduction is the oxidizing agent, because it's the one gaining those electrons.
Which leads perfectly into another vital give reason question. Why are oxidation and reduction considered concurrent processes? Why do they occur at the exact same time?
Because the scientific logic is perfectly balanced. You simply can't have one without the other. The exact number of electrons lost in the oxidation process has to equal the number of gained electrons in the reduction process. Think about it. If an electron is handed off, someone else must be there to catch it.
Moving right into Section 5, Reaction Speeds and Concentration.
So, now that we actually understand how these reactions operate mechanically, we have to study exactly how fast they happen.
Let's visualize this on a timeline. At the very beginning of a reaction, at exactly 0 seconds, the concentration of your reactants is at its absolute maximum, 100%, and your products are sitting at 0%. As time passes during the reaction, the reactants steadily decrease while the products naturally increase. Finally, at the very end of the reaction, the reactants hit 0%, and your brand new products have reached 100%.
Let's apply this concept to concentration. Give reason why. Why is the combustion of steel scours, the wire pads used for cleaning pots, why is that faster in a jar filled with pure oxygen than it is out in regular air? It's essentially a microscopic game of bumper cars. Pure oxygen has a much, much higher concentration of oxygen molecules than the normal air around us. By increasing the number of reactant molecules in a given space, you're drastically increasing the probability of them colliding with the steel. And simply put, more collisions mean a faster reaction speed.
Finally, Section 6, Catalysts and Everyday Applications.
What if you need a reaction to go faster, or maybe slower, but you absolutely cannot change the ingredients? Well, this is where scientists use catalysts. The strict definition: a catalyst is a chemical substance that changes the rate of a chemical reaction without changing itself or being used up.
It honestly has almost magical properties. A catalyst jumps in, combines with the reactants to help the process along, decreases the energy needed for the reaction to happen, and then it just separates from the products completely unharmed at the end. Literally, no chemical change or decrease in mass occurs to the catalyst itself.
A really brilliant, real-world engineering mechanism that uses this is the catalytic converter in modern cars. It uses ceramic cells shaped exactly like a honeycomb, which drastically increases the surface area. Now, these cells are coated with incredibly expensive precious metals like platinum, palladium, or iridium. As harmful engine gases flow through, this immense catalytic surface area rapidly speeds up the treatment of these gases before they even leave the exhaust. It's quite literally cleaning the air we breathe.
To wrap up our master class, we promised to ground all this chemistry in reality. So, here are some highly practical, everyday applications of a super simple chemical you probably have in your kitchen: sodium bicarbonate. You can put a small amount in vacuum bags to kill that weird dust smell, or drop some in waste baskets to remove bad odors.
Oh, and pour it down your kitchen sink with hot water. It actually speeds up draining. You can even soak legumes in water with a pinch of it to decrease the bloating associated with eating them. But my personal favorite? Place tarnished silver tools in a bowl lined with aluminum foil, add boiling water and sodium bicarbonate, and then dry them. It instantly returns their brilliant luster.
It's amazing. And with that, we have successfully broken down the entire chemical reactions unit. But as you close your notes today, I want you to just take a look around your room. From the battery powering your device to the very air you are breathing right now. What invisible chemical reaction is keeping you alive at this very second? Keep analyzing, keep wondering, and you will absolutely master your exams. Until next time.
Welcome to The Explainer. I'm your host, and today we are bringing you an elite, totally exhaustive masterclass breaking down the mechanics of electricity and nuclear energy.
That's right, and I am so incredibly thrilled to be here because honestly, we're treating this as an absolute zero shortcuts deep dive. We're going to examine the core scientific concepts, and then we're going to completely unpack the underlying mechanisms. You know, the real how and why behind them.
Exactly. So whether you're prepping for a massive final exam or you just want to totally master the physics of our modern world, you are in exactly the right place. Okay, let's dive right into this.
So here is the syllabus for our chronological journey today. We are starting at the absolute micro level with the atom, then the fundamentals of electric current. From there, we'll figure out how to measure it and map out resistance and Ohm's law.
Right, and then we'll look at how circuits are actually powered. After that, we pivot entirely to the immense power of the nucleus, exploring radioactivity, and finally wrap the whole thing up with the vital applications and safety protocols regarding radiation.
All right, Section 1, the foundations of flow. Before we can measure or use electricity, we really have to understand why electrons are moving in the first place.
First up, let's look at the atom itself. An atom is electrically neutral, and that's because the number of positive protons sitting right there in the nucleus perfectly equals the number of negative electrons rotating around it. The nucleus itself, well, that's positive due to those protons.
Exactly. And this leads to a super crucial exam question you guys need to know. Give reason. Why are electric wires made of metals? The secret is entirely in the atomic structure. Metals are characterized by a weak attraction force between that positive nucleus and their outermost negative electrons. Because of this really weak grip, when we connect a metal wire to an electric source, an electromotive force is generated. This force literally pumps these loosely held electrons, causing them to move right through the wire. And that continuous flow of negative electrons through a conducting material, that is the exact definition of an electric current.
Okay, so now that we know what the flow actually is, let's move into Section 2 and figure out exactly how we quantify this invisible river of electrons.
Our very first metric is current intensity, which we represent with the letter I. The textbook definition here is the quantity of electricity, or electric charges, in coulombs passing through a cross-section of a conductor in 1 single second. Let's break down the math on this. It's pretty elegant. I equals Q divided by T. Current intensity is measured in amperes, the quantity of charge, or Q, is measured in coulombs, and time, T, is in seconds. The absolutely crucial point here is understanding the proportionalities. The relation between current intensity and the quantity of electric charge is direct. More charge flowing, higher current. But the relation between current intensity and time is inversely proportional. If it takes longer for that exact same charge to pass, well, your current intensity is inherently lower. So next, we must understand why the current moves from point A to point B. This brings us to electric potential difference, represented by a V. Think about a basic physical scenario here. What actually happens if you connect two conductors that have totally different potentials?
To answer that, let's use a perfect real-world analogy: heat transfer. Imagine you connect a really hot object to a cold object with a metal bar. Heat naturally flows from the higher temperature straight to the lower one until they completely equalize, right? Well, electricity works exactly the same way. The electric potential is the state of a conductor that dictates whether electricity will transfer from it or to it. Connect two conductors with different potentials, and the electric current will invariably flow from the high potential conductor to the low potential conductor. And if they have the exact same potential, the potential difference is zero and absolutely no current is going to flow.
Let's look at the actual math behind that potential difference. It's mathematically defined as the work done, which we measure in joules, to transfer a quantity of electric charge of 1 coulomb between two poles. So the formula is V equals W divided by Q. The measuring unit for potential difference is the volt. Now, let's analyze a really common what happens if scenario for your exams. What happens if the work done is doubled while the charge remains perfectly constant?
Well, because potential difference and work done share a direct relationship in our formula, the potential difference will also double. It's just pure predictable physics.
To measure these invisible forces, we use two entirely different devices. The ammeter measures current intensity in amperes, while the voltmeter measures potential difference in volts. But listen up, because here is a major exam pitfall you must absolutely avoid. Pay close attention to how these devices are connected in a circuit. The ammeter must always be connected in a series connection. The voltmeter, however, is always connected in a parallel connection across the specific component you are measuring. Mixing these two up is a classic error that will definitely cost you points. So lock that parallel versus series distinction into your memory right now.
All right, moving into section 3. Electrons don't just flow completely unimpeded. They face opposition. And this opposition is what we call electric resistance.
Resistance, which we measure in ohms, basically comes in two flavors, constant and variable. The absolute master of variable resistance is the rheostat. Physically, it consists of a metallic wire coiled around a porcelain cylinder with a copper bar and a slider.
And the mechanism of operation here is totally critical to understand. When you move that slider, you are literally changing the length of the wire that the current actually has to travel through. It is a direct relationship. Increasing the wire length increases the resistance. And because the current now faces more opposition, the current intensity decreases. Conversely, decreasing the length decreases resistance and boosts the current. The rheostat essentially gives you total, manual control over the circuit's flow.
Which brings us to arguably the most famous relationship in electrical physics, Ohm's law. It states the mathematical relationship where potential difference equals current intensity times resistance.
Yes, the famous V equals I times R mathematical triangle. Or if we rearrange it, resistance equals potential difference divided by current intensity. From this one single formula, we can derive all our textbook definitions. For instance, what exactly is an ohm? Well, it's the resistance of a conductor when the potential difference is 1 volt and the current is 1 ampere. By mastering this one simple triangle, you basically hold the master key to solving any basic circuit calculation.
Transitioning into section 4, we're shifting from the properties of the current itself to how we actually generate and link that power together.
We basically have two main sources of electric current: electrochemical cells, like the batteries you use every day, and electric generators, like dynamos. Batteries produce direct current, or DC, which has a constant intensity and flows in only one direction. Generators, however, produce alternating current, or AC, which varies in intensity and actually flows in two opposite directions. Now, if you are ever asked the classic question, give reason why is alternating current better than direct current, the answer is super decisive. AC is vastly superior because it can be transferred over incredibly long distances to light up entire cities. And crucially, it can easily be converted into direct current when you need it for smaller devices. DC, on the other hand, simply cannot be converted to AC.
And how do we actually wire these batteries together? Well, you have two choices, series or parallel. In a series connection, you link different poles together, so the positive pole of the first cell connects to the negative of the second, and so on. But in a parallel connection, you connect all the similar poles together. All the positives go to one point, and all the negatives go to another. The difference in the resulting electromotive force, or EMF, is just huge. For a series circuit, you're creating a battery with a really high EMF, which you calculate by adding them up. E equals E1 plus E2, or N times E1 if they are identical cells. The parallel connection, however, results in an EMF equal to just one single cell. So, simply put, you use series for maximum power and parallel when you want to lower but sustain power over time. Okay, section 5. Let's leave those outer electrons completely behind and dive deep into the atom's core. It is time for the nucleus unleashed. Inside the nucleus, we have positively charged protons packed really tightly together. But wait, basic physics tells us that like charges repel. So here is a vital give-reason question. Why doesn't the nucleus completely fly apart due to this massive repulsion? The answer is the nuclear binding force. This is a staggering amount of energy acting inside the nucleus that completely overcomes the intense repulsion force between those positively charged protons, binding the atomic components tightly together. This binding energy is the literal source of nuclear power.
But as we know, not all elements are stable. Enter the French scientist Henri Becquerel, who discovered the incredible phenomenon of radioactivity. Unstable elements, known as radioactive elements like uranium or radium, have an excessive number of neutrons, way more than required for stability. and this creates excess energy.
Right, and to achieve a more stable composition, the atom's nucleus undergoes a fascinating process. It spontaneously decays. Visualize this excess energy violently ejecting from the core, emitting invisible radiation in the form of alpha particles, beta particles, and gamma rays. This spontaneous, natural decay is exactly what we call natural radioactivity.
Humanity eventually learned how to trigger this phenomenon intentionally. This is artificial radioactivity, radiation released during intentional nuclear reactions. And this artificial radioactivity falls strictly into two categories. It is either completely controlled, like we see in the safe, highly beneficial uses within nuclear reactors, or it is uncontrollable, designed specifically for catastrophic destruction in military applications like nuclear bombs. Which takes us to our final act, Section 6, where we examine the real-world impacts of nuclear energy, the horrific disasters, and crucially, how we protect ourselves.
When it's controlled, nuclear energy is incredibly beneficial across so many diverse fields. In medicine, it treats and diagnoses cancer. In agriculture, it eliminates pests. Industrially, it helps discover tiny product defects. It even powers rockets for deep space exploration and assists in drilling for oil. But perhaps most notably, it's used for massive electricity generation. The mechanism here is brilliant in its simplicity. The immense thermal energy, the sheer heat from the nuclear reaction, is used to boil water, producing incredibly high-pressure steam. That steam then physically turns massive turbines, which operate electric generators to create the electricity we use every single day.
But the dangers of a mistake are apocalyptic. On April 26, 1986, an operating error in the Chernobyl nuclear reactor in Russia led to a complete core meltdown. The explosion released massive atomic clouds straight into the atmosphere. And here is a terrifying give reason mechanism for you. How did that radiation spread so far and infect the food chain? Well, the polluted clouds were carried by the wind across Europe. When rain eventually fell, it dragged the radioactive isotopes right down to the earth, contaminating the soil and plants. Cows and sheep ate those polluted plants, meaning their milk and meat became heavily radioactive. It perfectly illustrates how easily natural weather systems can spread radiation globally.
And the impact on the human body is devastating, categorized by exposure type. Exposure to a huge dose in a short time, like over just one single day, causes acute devastation. It destroys the spleen, the digestive system, and the nervous system. But the very first thing affected is the bone marrow, severely decreasing red blood cells and making the victim incredibly sick.
On the other hand, exposure to a small dose over a long period, like months or years, is far more insidious. It causes physical effects, like skin cancer. It causes genetic effects, actually altering sex chromosomes, leading to abnormal births. And finally, cellular changes, eventually destroying the hemoglobin's fundamental ability to carry oxygen in the blood.
To manage this extreme risk, radiation absorbed by the human body is measured in sieverts. The absolute safe dose limit for a professional radiologist working in a hospital is 20 millisieverts per year. But for the general public, it is just 1 millisievert. Protection requires rigorous engineering logic. Radioactive wastes must be buried deep underground, surrounded by incredibly thick cement layers. And we must carefully select areas far from fault lines and earthquakes to prevent the cement from cracking, and keep it completely away from underground water aquifers to prevent the radiation from seeping into the public water supply.
And that concludes our masterclass. We have journeyed from the weak attraction forces of a tiny metal atom all the way to massive, steam-turning nuclear reactors and the critical safety protocols of the sievert.
Exactly. You've now seen the explicit mechanisms behind formulas like Ohm's law and the profound realities of the atomic core. If you fully grasp the flow of electrons and the power of the nucleus we unpacked today, you are completely ready. Good luck on your exam, and as always, keep questioning the invisible physics that power our universe. So what will you discover next?
Welcome, everyone, to this exhaustive masterclass explainer on the main principles of heredity. I'm your anchor for today's session. My job is to lay out the fundamental concepts, the exact scientific definitions, and really get into why these topics matter from the revision sheet.
And I'm your technical explainer. We are treating this as a zero-shortcut, ultimate breakdown of every single genetic mechanism outlined in our source material. We're going to pull apart the rules that govern the very fabric of living organisms, and we are leaving absolutely no stone unturned.
Right, so here is our syllabus for today. We'll start with the fundamentals of genetics, then Mendel's first law revealed, solving first law genetics, assorting two trait pairs, DNA structure and gene mechanisms,
And finally, engineering and the human genome. If you want to absolutely master this topic for your final exam, we really have to follow this strict chronological progression. We're building from foundational vocabulary straight through to advanced genetic engineering. Let's jump in. Section 1, the fundamentals of genetics. Look, before we can even begin to analyze complex genetic equations, we have to anchor our discussion in the precise vocabulary of heredity. So, genetics is fundamentally the science that researches the transmission of traits from one generation to another by studying similarities and differences between parents and offspring. Now, there is an absolute distinction between two types of traits here. Hereditary traits are, by textbook definition, traits that are transmitted from one generation to another. We're talking hair color, eye color, and blood groups. Acquired traits, on the other hand, are those not transmitted from one generation to another, like speaking different languages or playing football.
Exactly, and the underlying logic separating those two is strictly biological versus environmental. Hereditary traits are hardwired directly into your biology. You get them from your parents. Acquired traits, conversely, depend entirely on environmental learning and external practice. No one is born knowing how to speak French or tackle a striker.
That perfectly brings us to Gregor Mendel. He is universally recognized as the founder of heredity because his scientific studies literally formed the absolute bedrock of genetics. Now, exam papers love to ask a classic give reason question right here. Why did Mendel select the pea plant specifically for his experiments? Well, there are 6 distinct mechanical reasons for that. First, it's incredibly easy to plant and it grows extremely fast. Second, its life cycle is remarkably short, so you get results quickly. Third, and this is a completely crucial physical mechanism, its flowers are hermaphrodite. That means they carry both male and female organs simultaneously, allowing them to perfectly control their own self-pollination. Fourth, humans can easily artificially pollinate them. Fifth, a single generation produces a massive yield. And finally, the plant possesses several pairs of easily recognized contrasting traits. And Mendel meticulously selected 7 specific contrasting traits to track. He looked at flower color, so red versus white, flower position, axial versus apical, seed color, yellow versus green, seed shape, smooth versus wrinkled, stem height, tall versus short, pod color, green versus yellow, and pod shape, swollen versus sinuous.
Now please heed my warning on this. A highly common pitfall for students on exams is confusing the seed traits with the pod traits. Notice carefully that the dominant seed color is yellow, but the dominant pod color is green. You absolutely must pay close attention to that strict distinction, or you are going to lose points for sure.
Okay, moving on to Section 2, Mendel's first law revealed. We are going to completely deconstruct the specific mechanisms of Mendel's very first foundational experiment. The exact aim of this experiment was to study the inheritance of one single pair of contrasting traits. Step 1, Mendel let pea plants self-pollinate for several generations. Step 2, he removed the stamens, which are the male organs, from the flowers before they became mature. Step 3, he performed cross-pollination manually and then tightly covered the stigmas of the pistils. Let's aggressively tackle the give reason logic behind these steps because cause and effect is everything in genetics. Why self-pollinate for several generations? Well, to absolutely ensure the genetic purity of the traits. Why remove the stamens? To guarantee the prevention of self-pollination. And why cover the stigmas after manual pollination? To perfectly block any unwanted cross-pollination from the wind or insects. The experiment had to be a closed, flawless system. Let's look at the precise observational data. When Mendel caused a pure yellow seed plant with a pure green seed plant, the entire first generation, 100%, were yellow seeds. The green trait completely vanished. He then let that first generation self-pollinate. In the second generation, the green trait suddenly reappeared, yielding a strict mathematical ratio of 75% yellow seeds to 25% green seeds. That's a perfect 3-to-1 ratio.
And this exact observation birthed the formal principle of complete dominance. It states definitively that the dominant hereditary trait will always completely override and appear in the first-generation individuals when 2 pure contrasting individuals are crossed. The recessive green trait didn't actually cease to exist, right? It was simply completely dominated and hidden by the yellow.
To explain this mathematical phenomenon, Mendel formulated 4 structural assumptions. First, traits are transmitted by hereditary factors carried by gametes. Second, every trait is controlled by 2 factors, one from the father, one from the mother. Third, these 2 factors separate when gametes are formed. And fourth, they aggregate again during fertilization.
Think of it like mixing paint. These factors aggregate during fertilization to determine the final color. If the two factors are identical, say two dominant tall factors or two recessive short factors, we call that individual homozygous or pure. But if you mix them, combining one dominant factor with one recessive factor, the individual is heterozygous. The dominant paint completely covers the recessive one, producing an impure dominant trait.
All of this culminates in Mendel's first law, the law of segregation of factors. The airtight textbook definition is this: when two individuals of any pair of hereditary traits are different from each other, only the dominant trait appears in the first generation, while the two traits appear in the second generation in a ratio of 3 dominant to 1 recessive. Let's unpack the micromechanics of that segregation. It literally means that paired genetic factors physically separate or segregate completely independently into individual gametes during reproduction. They carry their codes in total isolation before recombining during fertilization, and that is exactly what guarantees that pure recessive traits can remain perfectly intact and hidden for a whole generation. Section 3. Solving first law genetics. The crucial next step is knowing exactly how to translate that theoretical law into hard, solvable mathematics on your exam paper. So, to solve genetics problems, pure individuals carry matching factors, while hybrid individuals carry mixed factors. You must fluently speak the universal notation system used to map this out. Parents are marked with a capital P, gametes with a G, and generations with F1 and F2. And here is the absolute golden rule of genetic notation. The dominant trait commands a capital letter, and the recessive trait is exclusively represented by a small letter. Therefore, a pure dominant individual will always have 2 capital letters. A pure recessive will have 2 small letters. And a hybrid or heterozygous individual will always be written as 1 capital followed immediately by 1 small letter. Let's trace an actual mating diagram step by step. We start by crossing a pure yellow individual, capital Y, with a pure green individual, small y. During gamete formation, those paired letters segregate. When they combine in the F1 generation, every single offspring receives exactly 1 capital Y and 1 small y. Because capital Y is dominant, 100% of the plants exhibit the hybrid yellow seed.
Now, let's dissect the subsequent step. When you cross 2 of those capital Y, small y hybrids together, the Punnett square mathematics strictly dictate the F2 generation's outcomes. You will perfectly derive 1 pure yellow, 2 hybrid yellows, and 1 pure green. This mathematically proves exactly how that hybrid dominant combination emerges to create a flawless 3-to-1 ratio. It is a beautiful mathematical certainty.
Section 4. Assorting 2 trait pairs. We are elevating the difficulty now by moving beyond single traits to analyze the simultaneous inheritance of 2 distinct contrasting pairs. What happens if we mix a pure tall plant with red flowers and a pure short plant with white flowers? Well, just as before, the first generation is 100% dominant. All plants are tall and all plants are red. But when we allow that generation to self-pollinate, we are faced with a massive, complex mathematical grid. We no longer just get a simple 3-to-1 ratio. Decoding the outcomes of assorting 2 pairs reveals a highly specific F2 generation distribution: 9 tall red, 3 tall white, 3 short red, and 1 short white. This is the famous 9 to 3 to 3 to 1 mathematical ratio. This massive statistical experiment gave us Mendel's second law, the law of independent assortment of hereditary factors. It dictates that if 2 individuals are different in a pair or more of contrasting traits, the trait of each pair is inherited completely independently from the others. To make this completely clear, apply this independent assortment to human traits. Inheriting the dominant trait for curly hair is a biological mechanism entirely independent of whether you inherit the trait for brown versus blue eyes. The biological sorting of your hair texture has absolutely zero mathematical influence on the sorting of your eye color. They assort totally independently. Section 5, DNA structure and gene mechanisms. It is time to pivot this masterclass from classical botany into the microcellular universe of human DNA. We really need to zoom deep inside the cell nucleus, where we find chromosomes. These chromosomes chemically consist of a nucleic acid, DNA, bound tightly with protein. The DNA itself carries the hereditary traits, consisting of smaller functional segments called genes. Zooming in even further, what structurally builds a gene? Scientists identified that genes are constructed from tiny, fundamental structural units known as nucleotides. These nucleotides sequence together to form the ultimate genetic blueprint of a living organism. We must credit the scientists Watson and Crick for their structural discovery here. They built the definitive, universally accepted structural model of the DNA molecule.
To visualize this abstract concept, they proved that DNA is composed of two long strands coiled tightly around each other. The concrete real-world analogy they used is that of a spiral ladder. This specific, coiled, ladder-like architecture is what we formally call the double helix. It literally holds the codes of life in its twists.
But how does a static double helix actually execute a function? For that, we turn to the Nobel Prize-winning mechanism discovered by scientists Beadle and Tatum, who outlined the exact mechanism of gene action. Think of this as a vivid biological factory line. Step 1. Every specific gene gives a specific enzyme. Step 2. That enzyme is singularly responsible for triggering a localized chemical reaction. Step 3. That chemical reaction results in the precise formation of a specific protein. Step 4. That protein goes to work to make the specific hereditary trait appear, like painting an eyebrow or making hair curly. Gene to enzyme to reaction to protein to trait. It is a flawless unidirectional chain of command. Section 6. Engineering and the Human Genome. For our final chapter, we will bridge the gap between this theoretical cellular science and modern-day technological application. Over 500,000 people globally lose their sight every year due to severe malnutrition, particularly in populations depending exclusively on standard rice. The exams give reason for this widespread deficiency. Regular rice lacks provitamin A. To solve this, biotechnologists engineered a brilliant, precise mechanism. They modified the genetic structure of the rice itself, inserting foreign genes that naturally produce keratin. Once consumed, this keratin perfectly converts into vitamin A inside the human body. This staggering application of genetic engineering resulted in what is globally known as golden rice. This mastery over the genetic code culminated in the Human Genetics Genome Project, initiated in October 1990. The exhaustive aims were to map every human gene, identify the exact genes responsible for diseases like cancer and diabetes, determine mutation functions, and completely understand human biology. And the staggering resultant of this massive undertaking? They discovered that all human beings share an incredible 99% similarity in our DNA sequence. All the diversity in humanity, every single trait, every difference you see, comes down to a tiny fractional 1% difference. Which leaves us with this final, profound question to consider on your exam and beyond. If we are 99% genetically identical, how will mapping and mastering that fractional 1% difference completely revolutionize our defenses against harmful diseases, environmental toxins, and dictate the future of targeted medical treatments? It's a question that pushes the boundaries of absolutely everything we've decoded today. Thank you for joining us on this rigorous, zero-shortcut masterclass explainer through the principles of heredity. Keep analyzing, keep questioning the mechanisms, and we will see you in the next session. Welcome to today's explainer. We're stepping into a true masterclass right now. I'll be your anchor, guiding us through the conceptual foundations of the human endocrine system. And I'm joined by my co-host, who's going to act as our technical explainer to break down the exact physiological mechanisms.
Thanks for having me. We're going to provide an exhaustive, micro-detailed breakdown of this scientific revision material. We'll be analyzing every definition, every cause and effect relationship, and every clinical application, no shortcuts whatsoever. So get ready to zoom all the way in on the microscopic chemical messengers that literally dictate human life.
Here's our strict chronological roadmap. We're starting with an intro to chemical messengers, then moving to the pituitary gland and growth. After that, we'll examine the thyroid gland and metabolism, analyze the pancreas as a dual gland, cover the emergency and reproductive glands, and finally, look at genetic engineering applications. Let's get right to it. All right, Section 1, an intro to chemical messengers. So, from previous studies, we already know the nervous system organizes our vital organs, but scientists have definitively proved it doesn't work alone. That is where the hormone comes in. Academically speaking, a hormone is a chemical substance, or message, that controls and organizes most of the vital activities and functions in the bodies of living organisms.
Right, and the crucial mechanism to understand here is that these aren't just random chemicals floating around. They are highly specific organizational signals. The human endocrine glands secrete more than 50 of these unique hormones, and they do it in strictly limited quantities. It's all about maintaining perfect homeostasis.
Notice the anatomy of this delivery system. Endocrine glands are explicitly defined as ductless glands. They do not have their own anatomical pipes or tubes.
Exactly. You can think of them like wireless transmitters. Because they're ductless, they secrete their chemical messages directly into the bloodstream, which then carries those signals to distant target cell receivers anywhere in the body.
Now, that brings us to a classic high-stakes give reason exam challenge. Why is blood considered the absolutely only way for hormones to reach their target cells?
To answer this scientifically, you have to understand the spatial relationship inside the body. You can't just memorize the answer, you've got to understand the geographical problem the body's actually trying to solve here.
The precise answer is twofold. First off, endocrine glands are completely ductless. Second, the target cells, the specific cells those hormones need to affect, are located far away from the gland doing the secreting.
Spot on. Because of this vast physical distance on a microscopic scale, combined with the lack of dedicated ducts, the constantly circulating blood vessel network becomes the mandatory, absolute sole transport highway for these chemical signals. It's literally the only route available.
Before we examine the specific glands, we have to define pathology. A hormone disorder is scientifically defined as the increase or decrease in the secretion of hormones when the endocrine gland that secretes it doesn't act properly. It's a very precise tipping point of imbalance. And as we move forward, keep in mind that pathology goes in both directions. It's a proportional relationship of error. Secrete too much, you get one severe disease. Secrete too little, and you get a completely different, often totally opposing severe disease. Moving to section 2, the pituitary and growth. The master gland. The pituitary is a tiny pea-sized gland located right below the brain, and it secretes the growth hormone, which determines a person's adult height. But look at the direct contrast when disorders happen. Let's analyze the mechanics here. Both of these trigger during childhood. Dwarfism is caused by a decrease in growth hormone secretion, which results in the body just stopping its growth. Conversely, gigantism is caused by an increase in secretion during childhood, resulting in continuous growth in the limb bones. The physical outcome of your entire body is directly tied to the exact volume of this single chemical during those developmental years. Section 3, thyroid and metabolism. Let's look at energy regulation. The thyroid, which sits right in the front of your neck, is the core engine of human metabolism. But this brings us to our next critical scientific logic check. Why must your daily food intake contain iodine?
This isn't just some casual nutritional suggestion. It is a rigid chemical necessity. Let's break down exactly why the thyroid gland will physically fail to do its job without this specific element.
The reason is that iodine physically enters into the chemical structure of the thyroxine hormone itself. If your food lacks iodine, it leads to a decrease in thyroxine secretion. We call this specific disorder simple goiter, and it's characterized by the enlargement of the thyroid gland in the neck.
And we absolutely must contrast that with its opposite, exothelmic goiter. This one's caused by an increase in thyroxine secretion in large amounts. The mechanism here accelerates metabolism so drastically that the symptoms include enlargement of the gland, accompanied by severe weight loss, nervous tension, and exophthalmosis, which is the scientific term for bulging of the eyes. Advancing to section 4, the pancreas, our dual gland and blood sugar balancer. Tucked right between the stomach and small intestine, the pancreas operates like a biological chemical thermostat. It secretes two totally opposing hormones, insulin, which reduces glucose sugar, and glucagon, which raises it.
Let's look at the cellular mechanism. When your glucose is too high, insulin stimulates your body's cells to use it for energy. Crucially, it also stimulates liver cells to store the excess glucose as glycogen. Now, when glucose dips too low, glucagon deploys to do the exact opposite. It stimulates the liver to convert that stored glycogen right back into glucose and release it into the bloodstream. It's brilliant. The liver is operating as a dynamic, chemically controlled storage bank.
Which leads perfectly into our next major exam question. Give reason why is the pancreas scientifically classified as a double function or mixed gland?
Students get tripped up by this all the time. It's not simply because it secretes two different things. It's actually about the very specific structural relationship between those two secretions.
Right. The exact scientific reasoning is that the pancreas secretes both insulin and glucagon, where the function of each hormone directly contradicts the function of the other.
Exactly. And when this contradicting balance fails, specifically if there's a decrease in insulin secretion, we see the disease diabetes. The mechanical failure here is staggering. Without enough insulin, body cells are literally unable to use the glucose. The sugar backs up, increasing blood glucose levels and spilling over into the urine. That's what triggers the classic symptoms of feeling continuously thirsty and needing to urinate frequently.
Onward to section 5, emergency and sex glands, the adrenal and reproductive systems. Let's map out how our biological alarm system works. The adrenal glands are located right above the kidneys, and they trigger a rapid chronological chain reaction when you're exposed to fear or horror. It happens in 4 incredibly fast steps. Step 1, the initial psychological exposure to fear. Step 2. The brain signals the pituitary gland. Remember, that's our master controller, which rapidly secretes an activating hormone into the blood. Step 3. This signal hits the adrenal glands, commanding them to instantly dump adrenaline. Step 4. The adrenaline floods the system, forcing organs like the heart and lungs to hyperaccelerate to manage the emergency. It is just a stunning cascade of chemical dominance.
Following the adrenals, we need to categorize the reproductive glands. In males, the 2 testes glands secrete the testosterone hormone, which is directly responsible for the appearance of males' secondary sex characters. In females, the 2 ovaries glands are responsible for 2 distinct chemical messengers. First, the estrogen hormone, responsible for the appearance of female secondary sex characters. Second, the progesterone hormone, which serves a highly specific structural function. It promotes the growth of the endometrium, which is the lining of the uterus, meticulously preparing the body for potential reproduction.
And finally, Section 6, Genetic Engineering, Curing Dwarfism.
This is where theoretical biology meets real-world, life-changing technological intervention. Let's look at the exact scientific process of reproducing human growth hormone to cure dwarfism. Let's analyze the sheer mechanics of this solution. First, scientists separate a human gene carrying the actual instructions for growth hormone. Second, they extract plasmid DNA from a bacterial cell. Third, they splice, or essentially insert, the human gene directly into that bacterial DNA. Fourth, they introduce this newly combined DNA back into the bacterial cell. Finally, the bacteria multiplies rapidly, synthesizing vast quantities of the human hormone. It's then separated, purified, and injected into dwarf patients.
The historical timeline of this application is pretty stark. Prior to 1979, scientists could only treat dwarfism by extracting growth hormone from the newly dead corpses of children.
And that method was deeply, deeply flawed. The amounts they could extract were microscopic, and it carried a massive risk of containing microbes that caused severe infectious diseases. But 1979 marked the absolute breakthrough of the bacterial genetic engineering we just detailed, and by 1985, extensive research definitively proved that this engineered hormone was a completely valid and safe treatment, a total game changer.
The final conclusion of the text states it perfectly. Scientists succeeded in 1979 in producing sufficient amount of growth hormone by genetic engineering technology. This hormone succeeded to treat children of limited growth.
It's such a prime example of human ingenuity completely overriding a biological hormone disorder. By understanding the chemical message, isolating the genetic code, and literally using bacteria as a manufacturing plant, science cured a condition that was previously thought to be a permanent physiological absolute.
We have exhaustively broken down the anatomy, the logic, and the applications of the human endocrine system today, from the master pituitary gland all the way to the dual-function pancreas. As we wrap up this masterclass, I want to leave you with a final provocative thought. Knowing the exact delicate science of these tipping points, what microscopic chemical messages are quietly flowing through your own bloodstream, keeping your body in perfect balance right now?
Hey everyone, and welcome to this Masterclass Explainer. Today, we are diving headfirst into the ultimate science revision sheet. We're going chapter by chapter, chronologically, so you can completely master this material.
Absolutely. We are breaking down every single main concept, every scientific law, and all those definitions you need to lock in. I mean, we're literally talking about the rulebook of the universe here. Everything from tiny microscopic atoms to the invisible forces powering massive cities. Let's get right into it.
So, here's our roadmap for today. We've got 4 distinct scientific units to tackle. First up, Unit 1, Chemical Reactions. Then we're moving on to Unit 2, Electricity and Radioactivity. After that, it's Unit 3, Principles of Hereditary. And we'll wrap the whole thing up with Unit 4, the Endocrine System. All right, kicking things off with Unit 1, Chemical Reactions. You know, the universe doesn't operate at just one single speed, right? So we actually categorize chemical transformations into 4 distinct reaction paces. First, we have very fast reactions. Second, relatively slow reactions. Third, very slow reactions. And finally, too slow reactions.
Yeah, and to put that into real-world terms, a very fast reaction is like an instant explosive burst of fireworks. Boom, done. A relatively slow one would be the process of mixing oil and caustic soda to make soap. Very slow takes months. Think of a piece of iron slowly rusting outside. And too slow? Well, that takes millions of years, like the formation of petroleum oil trapped deep underground.
Now, looking at the actual mechanics of these changes, we have equations for things like thermal decomposition, which is basically just where heat breaks down compounds, and simple substitution reactions. A great example to know is the decomposition of sodium azide into sodium and nitrogen gas.
And that specific formula isn't just a textbook theory, folks. That is the exact reaction that inflates a car's airbag in literally 40 milliseconds to save your life during a crash. I mean, that's just pure science in action.
When we track the concentration of these reactions over time, which we measure in moles per liter, we see the reactants start at 100% and just gradually drop all the way down to 0. The products do the exact opposite, starting at 0 and climbing up to 100% as the reaction finishes up.
Right, and sitting right there in the middle of that whole process is the catalyst. The amazing thing about a catalyst is that it remains completely unchanged throughout the entire reaction. It speeds everything up, but it never actually gets consumed itself. Pretty cool, right?
So, there are 4 textbook factors that dictate how fast these chemical reactions happen. First is the nature of the reactants, like how ionic bonds react way faster than covalent ones. Then you have the concentration of the reactants, the temperature, because heating things up makes those molecules collide much faster, and finally, those catalysts we just mentioned.
A brilliant everyday application of catalysts is the catalytic converter in your car. They use incredibly precious metals like platinum, palladium, or iridium to drastically speed up the neutralization of harmful engine emissions. They clean up the exhaust before it ever even hits the air we breathe.
Moving right along to Unit 2, Electricity and Radioactivity.
To really master electricity, you've got to understand the strict mathematical relationships at play here. The first connection is between current, measured in amperes, the quantity of charge in coulombs, and time in seconds. The second formula links potential difference in volts, work in joules, and charge. And the third is the classic Ohm's law, connecting resistance in ohms, voltage, and current.
If that sounds like a lot, just think of it like flowing water to make it super intuitive. Voltage is basically the water pressure, pushing the flow. Current is the actual amount of water flowing through. And resistance? That's the size of the pipe trying to slow that water down.
We also need to contrast direct current, which flows constantly in just one single direction, from sources like dry cell batteries, against alternating current. Alternating current has a variable intensity and direction, and it's produced by generators. Oh, and we also look at series connections versus parallel connections. The reason alternating current is so crucial is that it can be transferred over incredibly long distances. That is exactly why it's used to power our homes. And those parallel connections, they are vital because if one path breaks, the electricity just flows through the other paths. That's what keeps the rest of your house lights on even if one single bulb burns out. But electricity isn't the only unseen energy we're dealing with. Discovered by Omri Becquerel, natural radioactivity is the spontaneous, automatic decay of unstable elements that simply have too many neutrons. Elements like uranium or radium do this naturally as they try to achieve stability. And then you have artificial radioactivity, which is human-made. We can actually control it for peaceful uses, like inside nuclear reactors to generate massive amounts of electricity or to treat cancer. Or it can be uncontrolled, like what we see in nuclear bombs. When it comes to absorbing nuclear radiation, we measure that in a unit called the sievert. And for the general public, the safe limit is extremely strict. We're talking just 1 millisievert per year.
That's because the biological effects of overexposure are devastating. A massive dosage over a very short time immediately damages the bone marrow, the spleen, and the nervous system. But listen to this. Even a tiny dosage spread over a long period causes grim cellular and genetic mutations. It literally alters your chromosomes and leads to severe conditions like skin cancer.
All right, let's shift gears completely. Unit 3, Principles of Heredity. The founder of heredity, Gregor Mendel, is the one who figured out the fundamental rules of inheritance. His law of segregation is perfectly visualized using a Punnett square. When you cross 2 hybrid plants, meaning they carry 1 dominant gene and 1 recessive gene, the offspring follow a very strict mathematical probability. Exactly, because the dominant trait will always mask the recessive one. Because of that mechanic, 75% of the offspring will show the dominant trait, and only 25% will show the pure recessive trait. That right there is the famous 3-to-1 ratio. It is the absolute foundational mechanic of genetics. Later on, the scientists Bedell and Tatum discovered exactly how a single gene actually creates a physical trait, and it happens through a strict 5-step sequence. Step 1, the gene secretes an enzyme. Step 2, that enzyme triggers a chemical reaction. Step 3, the reaction produces a protein, and Step 4, that specific protein manifests the trait.
It is literally a biological factory blueprint. It's this incredibly precise, microscopic assembly line that builds an organism from the ground up.
Now, these genes are housed on DNA, which Watson and Crick famously modeled as a double helix shape. Today, thanks to the Human Genome Project, we know that more than 99% of human DNA is completely identical across all of us.
And scientists are using this deep genetic knowledge to save lives. For instance, they've engineered genetically modified rice to contain genes that produce keratin, which the human body then naturally converts into vitamin A. It's a massive medical breakthrough that fights malnutrition and prevents thousands of cases of childhood blindness.
Finally, we arrive at Unit 4, the endocrine system. For this unit, there are 5 major endocrine glands you really need to memorize: the pituitary gland, the thyroid gland, the 2 adrenal glands, the pancreas, and the reproductive glands.
I have to say, the pituitary is absolutely fascinating. It's known as the master gland. Not only does it secrete growth hormones, but it acts exactly like an orchestra conductor, constantly regulating and commanding all the other ductless glands in your body by dumping hormones right into the bloodstream.
But hormone disorders happen when this delicate system fails. For example, if the pituitary gland fails to secrete enough growth hormone during childhood, it results in dwarfism. But if it secretes way too much, that causes gigantism.
And we see similar extremes with the thyroid. A mechanical failure, like simply not having enough iodine in your diet, decreases thyroxine and causes a simple goiter, which is an enlargement of the neck. On the flip side, an oversecretion of thyroxine causes exophthalmic goiter, which leads to extreme weight loss and visibly bulging eyes.
Then we have the pancreas, which is this incredible mixed gland performing a beautiful biochemical tug of war. If your blood sugar gets too high, it secretes insulin, which steps in to pack that extra glucose away safely into your liver as stored glycogen. But let's say your blood sugar drops way too low. Well, the pancreas jumps back into action, this time secreting glucagon. It does the exact opposite, marching over to the liver, unpacking that stored glycogen, and dumping it straight back into the bloodstream as glucose. It is a perfect balancing act.
And that officially brings us to the end of our Masterclass Explainer. We really hope this structured deep dive leaves you feeling confident, totally inspired, and completely ready to absolutely ace those exams. Keep questioning and keep learning.
We've traced the very rules of existence today, from the different speeds of chemical collisions and the mathematical flow of electricity, all the way down to the 3-to-1 ratios of our genetic code and the delicate hormonal balances that literally keep our bodies functioning. When you take a step back and look at how all these units connect, from microscopic atoms right up to the DNA that makes us human, isn't science just the ultimate playbook of life?
Welcome, everyone, to a very special high-energy explainer. I'm your host and resident science teacher, and today we're taking your Prep 3 Science Final Revision sheet and transforming it into the ultimate gamified exam challenge.
And I'm your interactive learner and co-host. I've actually put my normal flashcards away, my brain is completely warmed up, and I am so hyped to tackle this material. If you're listening out there, you are about to get a massive upgrade to your study session.
Love that energy. We're going to rapid fire through the most critical definitions, the absolute trickiest give reasons, and those classic exam traps. The goal? So you can walk into that test with total confidence. So, the gauntlet has officially been thrown down. Are you ready to completely crush your science final?
Oh, I was born ready. But, I do realize this revision covers just a massive amount of ground. We've got complex equations, laws of physics, human biology. It's all mixed together.
Exactly, which is why we're gamifying this explainer. I'll be pitching the absolute toughest questions, and you, my friend, are going to help us break down the answers. All right, here's our revision roadmap. We have 4 distinct challenge levels to conquer today. Level 1, Chemistry Chaos. Level 2, Powerful Physics. Level 3, Biology Basics. And finally, Level 4, the Lightning Round.
4 levels to total science mastery. Sounds like a solid plan. Let's not waste any time.
Level 1, Chemistry Chaos. We're focusing heavily on reactions and rates here. All right, let's get into it. Hit me with the first challenge. I know the exam always loves to test us on why some reactions happen in the literal blink of an eye, while others just seem to take forever. Can you break down that difference for me?
I certainly can. Let's highlight a really classic exam trap here. When we compare reaction speeds, ionic compounds are incredibly fast. Why? Because they easily dissolve and dissociate into free ions in water. Okay, so because they break down into free ions right away, they're just ready to react instantly? Spot on. On the flip side, covalent compounds are much slower. They don't break into ions easily at all. The reaction actually requires breaking the physical bonds between molecules, which obviously takes significantly more time. Next up, a absolutely crucial definition. What do we call a substance that changes the speed of a reaction without actually being consumed by it?
Oh, that has to be a catalyst.
You got it. Think of a catalyst kind of like an enthusiastic cheerleader on the sidelines of a basketball game. They're hyping up the players, speeding up the momentum of the whole game, but they never actually step onto the court to shoot the ball. Some great examples are manganese dioxide, which speeds up the decomposition of hydrogen peroxide, or even the enzymes in sweet potatoes that act as natural biological catalysts. Let's test a practical application of these super fast reactions. Here's a rapid-fire question for you. What inflates an airbag during a sudden car collision? I know this one involves a rapid chemical decomposition to save lives, but what exactly is inside the bag? Oh, wait, it's nitrogen gas.
Correct.
During an accident, a rapid chemical reaction triggers and instantly fills the airbag with nitrogen gas, or N2. It's just a perfect real-world example of a life-saving, ultra-fast chemical reaction. Before we leave chemistry chaos, we really have to tackle the biggest mix-up on the entire exam: oxidation versus reduction. So, oxidation is the process where an atom loses electrons or gains oxygen.
And reduction is the exact opposite of that, right? It gains electrons or loses oxygen.
Yes, the exam will definitely try to trick you with these definitions. Just remember, they are complementary processes. If one substance loses an electron, which is oxidation, another substance literally must gain that exact same electron, and that's reduction. Boom! Level 1 is complete. Hey, don't get too comfortable, because we're moving right along to level 2, powerful physics. Imagine we're looking straight inside a glowing, high-tech circuit box. We can see all the major components working together: the battery, the resistors, and the meters.
It honestly sounds like something out of a sci-fi movie. But the exam always asks us to identify these specific meters. How do we actually tell the ammeter and the voltmeter apart when we're building a circuit?
So, the absolutely crucial point is how you connect them. Here's your ultimate cheat sheet rule. The ammeter is always connected in series. It forces all the electrical charges to flow straight through it to measure the current intensity, which is in units called amperes.
Got it. And the voltmeter?
The voltmeter is connected in parallel, creating its own separate little branch. It measures the potential difference between 2 points in volts. Just remember, ammeter in series for current, voltmeter in parallel for potential difference. Don't mix those up. Now, what ties potential difference and current together? That would be the legendary Ohm's law. The formula is R equals V divided by I. So resistance equals potential difference over current. Exactly right. Think of potential difference, or the voltage, as the pressure pushing the electricity through the wire. The current is the actual physical flow of those electrons, and resistance, which is measured in ohms, is the opposition fighting that flow. Ohm's law shows us they're all perfectly mathematically linked as long as the temperature is constant. Let's pivot away from electricity and look at the forces inside the nucleus. Here's your next definition challenge. What's the scientific term for the spontaneous decaying of an atom's nuclei in certain elements just to reach stability? Oh, that is radioactivity. And it was discovered by Henri Becquerel, right? Perfect score. And remember, radioactivity can be completely natural, like from radioactive rocks or cosmic rays in space, or it can be artificial, like the controllable reactions we use in nuclear reactors, or the uncontrollable ones in nuclear bombs. All right, let's jump to level 3 and see how all this builds into our understanding of life itself. Welcome to Biology Basics. We're officially trading wires and chemicals for DNA and glands. Yes, my favorite subject. Bring on Gregor Mendel and his famous pea plants. You asked for it. Mendel showed us exactly how hereditary traits are passed down through dominant traits versus recessive traits.
Right, so a dominant trait is kind of like a loud voice in a crowded room. It completely covers up the quieter recessive trait.
That's a fantastic analogy. Dominant traits, like being a tall pea plant or humans having free earlobes, appear 100% of the time in the first generation. But those quiet recessive traits, like dwarf plants or attached earlobes, don't just vanish forever. They sneakily reappear in about 25% of the second generation. Moving from genetics to how our body actually communicates right now in real time. What is the definition of a hormone?
A hormone is a chemical substance that controls and regulates the functions of most body organs, and they're secreted directly into the bloodstream by our endocrine glands.
And this just brilliantly illustrates how our body sends long-distance instructions. Hormones are basically chemical text messages. They bypass the normal ducts, jump straight into the blood highway, and travel to specific target cells far away to deliver their regulating instructions.
Let's rapid-fire the major senders of these chemical text messages. The pituitary gland is the master gland because it controls the other glands. The thyroid secretes thyroxine to regulate food assimilation. Oh, and the pancreas is that super cool double-function gland. It secretes insulin when your blood sugar is too high and glucagon when your blood sugar drops too low. You are on fire today. And definitely don't forget the adrenal glands, which pump out adrenaline to prepare your body for sudden emergencies.
Okay, we've conquered chemistry, physics, and biology. But the exam always throws in those tricky give-reason questions at the end. It's time for the final level, the lightning round. My palms are a little sweaty, but I'm ready. Let's do this flashcard style. Here's your first lightning round question. Give reason. Why is alternating current, or AC, preferred over direct current, DC?
Ooh, because alternating current voltage can be easily stepped up or down using transformers, and it can be transmitted over incredibly long distances without losing much power. Direct current just can't travel that far efficiently.
Flawless answer. Absolutely right.
Okay, final question of the explainer. It's a really tricky biology trap. Give reason. Why does learning to walk not count as a hereditary trait?
Ha, nice try, but I won't fall for that trap. Learning to walk is an acquired trait. It's developed through lifetime practice and experience. It is absolutely not passed down through the genes in our DNA.
You absolutely nailed it. Acquired traits stay with you, but hereditary traits are the ones actually written in your chromosomes.
And just like that, we've reached the end of the Ultimate Prep 3 Science Exam Challenge. You've survived the chemistry chaos, mastered the electrical circuits, and decoded human biology. This was an incredible revision session. I feel so much more prepared just hearing how all these concepts connect together. Well, that's the real power of structured learning. We wish you the absolute best of luck on your science exam. Keep observing, keep questioning, and ask yourself, what scientific mystery will you solve next?
Yalla bina ya shabab. Welcome everyone to the Ultimate Prep 3 Science Exam Night Bootcamp. I'm your host and resident science teacher, and we're here to make sure you are absolutely 100% ready for tomorrow's exam. And I'm your co-host, ready to play the ultimate revision game. If you're looking for a fast, high-energy explainer session to lock in all those tricky definitions, equations, and give reason questions from your revision sheet, well, you're in the exact right place. Let's get this bootcamp started.
Okay, let's dive right into this. The rules for tonight's session are simple. We are going rapid fire. I'm going to throw the most common exam traps, questions, and concepts straight at you.
And I'm going to try to answer them before the students listening at home can. We're breaking down the toughest concepts so they actually stick. No more memorizing without understanding. We're going to crush this exam.
All right, Section 1, Physics Concepts and Exam Traps. Let's dive straight into the deep end with electricity. So, think about an electrical circuit setup. We've got 3 main devices you absolutely need to know: the ammeter, the voltmeter, and the rheostat. Rapid fire time. What does each one actually measure?
Okay, easy. The ammeter measures current intensity in amperes, the voltmeter measures potential difference, or electromotive force, in volts, and that slider thing, the rheostat, that controls the variable resistance in the circuit.
Spot on. But knowing what they do is really only half the battle. How do we actually connect them? So the crucial point here is the connection, ammeter versus voltmeter. Right, the ammeter goes in series, tawali, and the voltmeter goes in parallel, tawazi. But honestly, sometimes I totally mix them up under exam pressure. Well, here's a simple analogy to really make it stick. Think of a series connection like students holding hands in a single line. The current literally flows through everyone, one by one. That's your ammeter. It has to be right in the line to count the flow. But a parallel connection, that's more like a giant group hug around the teacher. The voltmeter connects across the component, branching off to measure the difference between 2 specific points.
Let's move to Ohm's law and see how this builds. Resistance equals voltage divided by current. Now, I'm flagging a huge exam trap right here. Listen closely.
All right, I'm ready. Lay it on me.
If you double the current passing through a fixed 5 ohm resistor, what happens to the resistance? Does it double to 10 ohms?
Oh, no way, I know this one. It's the total trick. The resistance remains exactly 5 ohms. It does not double. The resistor is a physical piece of hardware, right? Its resistance is totally fixed. If the current doubles, it's just because the voltage doubled, not because the resistor magically changed. Exactly. Do not fall for that tomorrow, guys.
Next up, electric current sources, AC versus DC. Where do they even come from?
So cells and batteries produce direct current, or DC. It's got a constant value and direction. But dynamos, like electric generators, those produce alternating current, AC.
And here's a classic give reason question for you. Why is alternating current preferred over direct current?
Because AC is just way more flexible. You can step it up or step it down, and it can be transmitted over incredibly long distances without losing much energy. DC can really only go short distances.
Moving right along, Section 2, Equations and Hormones. The lightning round begins now.
Let's go! I am so ready to keep up with these rapid-fire chemistry and biology facts. Hit me.
Warning tone. This is a classic mix-up on literally every exam. Oxidation versus reduction. Define them for me, specifically by electrons.
Okay, so oxidation is a chemical process where an atom loses an electron or more. Reduction is when a substance gains an electron or more during a reaction.
Exactly. Oxidation is losing, reduction is gaining. Or, if we're talking oxygen, oxidation gives oxygen and reduction takes it away.
Next, thermal decomposition, or as we say, in halal Harari. What exactly happens when you heat red mercury oxide? Give me the equation. Oh, I can see it in my head. 2HGO, we add heat, the little triangle symbol over the arrow, and it breaks apart into 2Hg, which is a silvery precipitate, plus O2 gas, which evolves and escapes. Perfect. The red turns to silver and oxygen gas is released.
Okay, give reason. Magnesium replaces copper in its salt solutions, but copper absolutely cannot replace magnesium. Why? Because of the chemical activity series. Magnesium is way more chemically active than copper. So, when you drop magnesium into blue copper sulfate, the magnesium basically kicks the copper out, taking its place to form magnesium sulfate, and the copper just falls out as a red precipitate.
Brilliantly explained. It is all about who is higher up on that activity series.
Switching gears to biology now, endocrine system rapid match. I'm going to name the gland and you give me the hormones and their function. Ready? Pancreas secretes insulin to lower blood sugar by storing it, and glucagon to raise blood sugar by releasing it from the liver. Thyroid gland secretes thyroxine, which regulates food assimilation and releases energy, and calcitonin, which totally controls the calcium levels in your blood. Wow, you are seriously on fire today. So we just named all these hormones. But what actually is a hormone? Let's make absolutely sure this core definition is locked in.
Right. A hormone is a chemical substance or message that controls and regulates the functions of most of our body organs.
Exactly. It's literally a chemical message sent directly via the bloodstream. It's basically how your brain and glands send text messages to the rest of your body. All right, Section 3, Mendel's Genetics Masterclass. Let's review the founder of heredity, Gregor Mendel, and his legendary pea plants.
Oh yes, the guy who changed science forever. Let's lock in his core findings.
First up, what's the actual difference between a dominant trait and a recessive trait? Give me an example that humans have. Well, a dominant trait is the one that appears in all individuals of the first generation when you cross two pure traits. It dominates. A recessive trait is the one that completely disappears in that first generation. For humans, having a free earlobe is a dominant trait, while having an attached earlobe is recessive. Spot on. The dominant gene just completely overrides the recessive one if they're paired together. So the recessive trait disappears in the first generation. But does it stay gone forever? What percentage does the dominant trait appear in the F2 generation, the second generation of Mendel's experiments?
It totally comes back, but the dominant trait still wins overall at exactly 75%. That's Mendel's classic 3-to-1 ratio. 3 dominant for every 1 recessive.
That 75% number is pure exam gold, guys. Remember it. Now, for a major exam trick. Mendel was incredibly careful with his pea plants. Give reason. Why did Mendel remove the stamens from the flowers, and why did he cover the stigmas? Okay, so he removed the stamens, the male parts, before they matured to entirely prevent self-pollination. He wanted to strictly control who pollinated who. And then he covered the stigmas, the female parts, to prevent any random unwanted cross-pollination from the wind or insects. Perfect. Stamens removed equals no self-pollination. Stigmas covered equals no cross-pollination. Please do not mix those two up tomorrow.
All right, we've made it to section 4, the final pop quiz. If you're listening at home, try to shout out the answers before we do.
This is it, the ultimate simulation. Let's see what you've got.
Question 1. What happens when you connect two conductors of the exact same electrical potential with a wire?
Absolutely no current flows. Electric current only transfers when there's a difference in potential. If they're perfectly equal, nothing moves at all.
Question 2. Why are the chemical reactions of ionic compounds so much faster than covalent compounds? Because ionic compounds actually dissolve in water and totally dissociate into free, moving ions. Covalent compounds just stay as whole molecules, so their reactions require you to slowly break molecular bonds first. Basically, ions are fast and molecules are slow. Question 3. True or false? Mendel removed the stamens from pea flowers to prevent cross-pollination. False. I caught that trap. He removed the stamens to prevent self-pollination. Final question, we've got a surprise math problem. Calculate the voltage if the work done is 800 joules to move a charge of 40 coulombs. Go. Okay. The formula for potential difference or voltage is V equals W over Q, work divided by charge. So 800 joules divided by 40 coulombs, let's cross out the zeros. 80 divided by 4, that is exactly 20 volts.
20 volts is absolutely correct.
And that brings us to the end of our Ultimate Bootcamp Explainer. You have navigated circuits, balanced equations, unlocked genetics, and dodged every single exam trap we threw at you today. Yeah, if you could keep up with us today, you are going to absolutely crush that paper tomorrow. Get some rest, trust your preparation, and remember to read every single question carefully. From both of us, the absolute best of luck to all the Prep 3 students on your final science exam tomorrow. You've got this. Now, one final thought for you to ponder before you walk into that exam room. If energy cannot be created or destroyed, how will you channel all your existing energy into tomorrow's success? Go get 'em.